Hello and welcome back. This is the second part, second micro lecture for chapter 1. And in this one, we're still going to be looking at major themes in anatomy and physiology. But here we're going to be looking at human origins and pretty much how this book is set up. So we'll finish up with a couple other scientists that we're going to talk about later on in this semester. Again, they're not going to be in red, so it's not important. But there is a couple of course learning objectives in this lecture. So I'll point them out when we get to them. So starting with a couple of scientists, Charles Darwin, you probably all heard of. He wrote The Origin of the Species, and what he did with that book is he was the first one to write down about evolution. And he started looking at different types of birds, and these different types of finches were the same species, but they were completely different with their evolution. So he was able to say that because they grew up in different areas, they evolved differently, and he was drawing a line back through their evolution that they all came from one type of finch that then grew apart through millennia of being separated and being in different environments. Again, he wasn't the first one to have this idea, but he was the first one to write it down. So that's why when you think about evolution, most people are going to point to Charles Darwin. But it was a lot of English scientists and a lot of English noblemen that had the same idea during this time in the 19th century. So, theory of natural selection. So, Alfred Russel Wallace, a British biologist, shares credit with Charles Darwin for the theory of natural selection. So, these two people, but his book came out a couple months later, so you probably never even heard of that name before. So that's why I bring it up now. And it just looks at how species originate and change through time. They change the prevailing view of our origin, nature, and our place in the universe because we weren't always the same creatures that we were today. We evolved from something else, which means that thousands and hundreds of thousands of years, humans are going to look much differently than they do today. And by looking at evolution, evolution has to change from something. So in the 1800s, they didn't know what DNA was. So they were kind of thinking, okay, we evolved from something, but how did we do this? So we know now that change in our genetic composition of a population of organisms, that's what's actually causing it. Your DNA changes, the DNA is going to change the proteins being made, and the more proteins that are made can change how something looks, how something acts, or how something behaves. And you can look at a development of bacterial resistance to antibiotics. In hospitals now, you hear about antibiotic-resistant strep and superbugs in hospitals because of the use of antibiotics. They've learned how to live alongside antibiotics, and that makes them resistant to them. And also, AIDS can change because it's a virus. COVID can change over time. Any types of virus can start to change and mutate over time, and it's done that by its genes and by its cellular makeup or molecular makeup. And during this time, they also looked at natural selection. So some individuals within a species have better advantages over their competitors. Maybe they look different, and the way it looked can be better camouflage. Maybe it was brighter, and the brighter colors attracted more mates. And if you have more mates, or you live longer, you could then pass on your genes to more offspring. It might even have a disease resistance. So again, if you live longer, you can have more offspring. Those offspring will then be more beneficial to mate with more people or more animals over time. And then the more kids over time, that's how evolution would work. So you have adaptations and features of organisms' anatomy, physiology, behavior that have evolved in response to these selection pressures. But pretty much, if one species or one type of look or whatever is viewed to help out that animal, those genes can be passed on. So if we look closer at genes, our closest relative is a chimpanzee. So if you're thinking chimpanzee, all right, 1.6 of the DNA structure is difference between a human and a chimpanzee. And if you're thinking, so that means that your adenine or your adenosine, the A, G, C, and T's, those nitrogenous bases that we're going to discuss in chapter 4 in genes and cellular function, those little base pairs, only 1.6 is different between a human and a chimp. And if you think about this, the difference between a chimpanzee and a gorilla is 2.3. So there's a bigger gulf between the genes between a chimpanzee and a gorilla than a human and a chimpanzee. And that's not to think that we are chimps or we look like chimps. It's just the fact that when we go over chapter 4, it might not look like you change a one base pair. You might think that that's not important, but changing just 1 or 10 base pairs out of a gene that's 10,000 changes that function of that protein that it makes. And by changing it just a little bit, by either changing the gene or changing the base pair either by radiation or by copying it incorrectly, that's how evolution is going to occur and changes within a group. And then if you're looking at the study of evolutionary relationships, you can start choose which animal model would be better for biomedical research. So that's why a lot of them that do genes, they start with Drosophila. They only live 12 hours long, so you can get multiple generations within a week. You might take a mouse over a rat. You might do mice research if it's more genetic, because mice genes are more well known and they have a lot more models of different types of diseases in mice than they do in rats. If you're doing more behavioral or something like that, you might be using rat over a mouse, because if you've ever had a friend that had a rat as a pet, you can train a rat, which is kind of strange. You can't train a mouse, but you can train a rat. So just by knowing the genes and evolutionary relationship between these animals, you can pick a better model for your research. And walking upright, other Homo species discovered recently are still a matter of considerable debate, because they don't know if they are directly related to us or if they all kind of converged into one type. That's why they say there's a missing link. It's one thing that you'll probably never be able to, pin down, because Homo sapiens, which is what we are, originated in Africa around 200,000 years ago. And if you're thinking 200,000 years, what came before that? Bones break down, teeth break down, skeletons break down. So you don't have a lot of fossilized records of things that go back all those years of humans, and then trying to pick up fragments. Where are they, and are they our ancestors, or is it a direct link? It's kind of complicated. That's why it's a considerable debate between these. And one thing we started using right after Darwin is evolutionary medicine, trace some of our diseases from imperfections in our past. So we'll talk about different genetic things like Tay-Sachs. Tay-Sachs is a disease that kind of can be traced back to Eastern European Jewish populations, because their genes were passed on over time. And you can also look at sickle cell anemia. That is a trait that is very beneficial for one group of people, and then when they left that environment, it became worse, because they died because they would have blood clots and other blood disorders. But if you lived in a place with malaria, you'd probably die of malaria before you died of a blood clot or a stroke. So it's one of those things where all these evolutionary issues can make it more difficult for you to pass on your genes. But on this page, Homo, same, equal, or like. The prefix Homo, same, identical, or equal. It's derived from the Greek word homeos, And that's going to bring us to homeostasis. That's homo habilis. Homo sapien means same. So every time you see the word prefix of homo, it's going to mean same anytime we talk about it in this class. And the main thing we're going to be talking about later on is going to be homeostasis, which is going to keep things in one state or a balanced state. So let's look at how this textbook is made up. So your textbook, we're talking about the history right now. We're going through all this. Next chapter is going to be the chemistry of life, where we're going to talk about what makes up an atom, the neutrons, protons, and electrons. How do those atoms come together in molecules? How do they become bigger molecules? Then in chapter 3, we're going to look at the cell. So the cell is going to be made up of a lot of different types of organelles. And in chapter 3, the cell is the basic building units of life. All living things are composed of cells, and cell is the smallest thing that can create life. You're like, okay, because a cell, if it's in the correct environment, in a petri dish or whatever, it can create, it can grow, and it can continue to do whatever its function is supposed to be. And then in chapter 5, I believe, chapter 5 is going to be human tissues, and then we'll start looking at what the tissues look like, where are they found in the body, and what are they doing in the body, so their function. And then we start looking at bones, muscles, and sense organs, nervous tissue, the brain. So we start looking at organs. The brain is an organ. AMP2 is just going to be looking at organs like digestive system, circulatory system, reproductive system, respiratory system. All of these things are going to be in AMP2. And then finally, at the end of AMP2, after taking this class and getting a good background, you're going to be able to figure out how an organism works. So your book starts off at atoms in chapter 2, and by chapter 16, you understand how everything works together. So what is this and why is this important? Why are the labs important? The labs are important for you to do dissections. You're supposed to look at a cow eye or a sheep eye or a brain of a calf, or in AMP2 when you look at the cat and do dissections. It's important to do this because of this one term, anatomical variation. No animal, but no importantly for you, because it's human anatomy, no two humans are exactly alike. 70% have very common structures. 30%, they're different. So let's think. Normal kidney structure, you're going to have a kidney on the right side. This is the left, that's the right. It's going to be a little bit lower because the liver is going to be right there. On some of them, the right side is going to be not joint to the adrenal gland. It's going to be all the way down here. Some, like 3%, it's going to be a horseshoe kidney. And if you're wondering why that's important, if you're a doctor or a nurse doing surgery, you go in and you see this, and you don't know about something called the horseshoe kidney. You don't know about all the different types of anatomical variation for different organs or groups or organ systems. You see this, you're not going to know how to react. So by knowing anatomical variations, but seeing them right now, to just be aware that things are not always what they look like in the textbook is very, very, very important. AMP2, you talk about this. This is a human normal branches off the aortic arch, where you have the brachiocephalic, left common carotid, and then the left subclavian. Sometimes the left common carotid is coming off of the brachiocephalic. Sometimes it doesn't come off the brachiocephalic. Sometimes it branches off the left subclavian. Sometimes the right common carotid is also coming off the aortic arch. It's the right common carotid, the left common carotid, and the left subclavian. By knowing this, so if you see this in imaging, where you see it on a scope or a surgery, by knowing these things exist, you can be able to deal with them in a more calm manner and be able to work in a better frame of mind by helping whatever the patient originally went in for. So anatomical variation is very important. But now we're going to look at something in red. So homeostasis and the negative feedback. So homeostasis is the body's ability to detect change, activate a mechanism that opposes it, and thereby maintain relatively stable internal conditions. That might not seem that important to you. But think about it if you have ever seen a yin and yang. That philosophy pretty much means that if things go to one side, they're going to balance off on the other side eventually. Calm, peace, all these other things are maintaining a state of homeostasis. If you look at homeostasis, homeostasis uses the prefix homeo, meaning similar or same, so same state. You keep it at a stable state. Claude Bernard in the 1800s, mid-1800s really, he constant internal conditions regardless of external conditions. He's the one that started to do internal body temperature. Even if it was really cold, your internal body temperature stays the same. And he was the first one to kind of think, well, there must be some process in the body that allows us to keep it at that internal state. And it was a few years after that, half a century after that, that Walter Cannon coined the term homeostasis. And he's the one that kind of found there's something called the negative feedback. And it keeps a variable close to a set point. And if you lose homeostatic control, illness or death. Because when we talk about negative and positive feedbacks, which are going to be important, these are all going to be thoughts for learning outcomes for this course. Negative and positive feedback. So let's look at negative feedback. So a negative feedback keeps it at a set point. So it doesn't get too high, doesn't get too low, it keeps it at a set point. And if you kind of think about it, it's like your thermostat or your air conditioning at home. If your room temperature, this is a thermostat. I know it's kind of weird to see this, a thermostat in Florida, but that's what the book has. So if the room temperature falls to 66, it's going to activate the thermos to heat it back up. It heats the room up to 70. It causes the thermostat to shut down the furnace. The room will cool down, and then when it cools down too much, it turns the furnace back on. That's why throughout the day you'll hear the air conditioning kick on, then shut off. Kick on and shut off because your thermostat is set at a set point. If the air goes on too long, it goes right below it. And then when it's shut off, it'll raise back up. Then it will turn on, and it will be almost like a wave. So let's look. This is a feedback loop where you turn the furnace on, heats up the room, you shut it off, the room gets colder, then it goes back on, room gets colder, and then that's your feedback loop. It doesn't stay at a set point, but it averages at a set point. Another way to think about it for you is sweating or shivering. So you want to keep, these are just two things our body uses to keep us either colder or hotter. If it's too hot, we'll sweat. If it gets too cold, we'll shiver. If it gets too hot, we'll sweat. Not pretty much like that, but usually sweating will cause evaporation. Evaporation causes cooling. Shivering will warm up the muscles and warm up the blood in those muscles, and that's how shivering will warm up the body and sweating will cool off the body. But if it's too warm, vessels dilate, vasodilation in the skin, sweating begins to cool off the body. If it's too cold, vessels in the skin constrict and shivering begins. Shivering will create heat. The heat in the muscles will start warm the blood. And that's a negative feedback. So another homeostasis and negative feedback. Sitting up in bed causes a drop in blood pressure in the head, in the upper dorsal region. So it's a local imbalance in homeostasis. So if you stand up too quickly, you get lightheaded. We've all done that. And it's detected by baroreceptors in your carotid bodies just above your heart, in the lower part of your neck. So this pressure changes in this chemical imbalance. These baroreceptors are these nerve endings in the arteries near the heart. They alert the cardiac center in the brainstem, then they transmit to the cardiac center. And you're like, okay, if you stand up too quickly, you get lightheaded. The brainstem then tells the heart to beat more blood, more oxygen to the brain, and then you're not as lightheaded anymore. So that's an example of a negative feedback. Positive feedback is opposite. So positive feedback is a self-amplifying cycle. So this means it only goes in one direction. So if you have a fever, that's an example of a positive feedback loop, because a fever will stimulate the digestive system to digest more food. Digesting more food creates more heat. More heat will raise the fever up. Because your fever is hotter, it's going to stimulate the digestive center to digest more food, more heat, fever goes up, and that leads in one direction. The feedback loop is repeated. Change produces more of change in that one direction. And it's a normal way of producing rapid changes. So childbirth, you've all heard of how close the contractions are. The contractions are controlled by a positive feedback loop. Because they're getting closer and closer together, that means the baby's going to be born sooner. Blood clotting the same way. You don't want blood to keep leaking out of a vessel or your body, so blood clotting is also positive feedback. Protein digestion, fever, and generation of nerve signals, these are all going to be self-amplifying cycles because you want them to keep going in one direction. You don't want it to stop after a while. So let's look at childbirth. So during childbirth, the head of the fetus pushes against the cervix, and that stimulates its nerve endings. The hormone oxytocin is secreted from the pituitary gland. Oxytocin travels through the bloodstream to the uterus, stimulating it to contract. This action pushes the fetus downward towards the cervix, thus stimulating the cervix more, causing positive feedback loop to be repeated. If you're like, what? Of course, there's a picture. So first, the fetus is going to push on the cervix. The cervix is then going to send a signal to the pituitary gland to release oxytocin. Oxytocin then causes the uterus to contract. The contraction of the uterus pushes the fetus down, the head pushes on the cervix, and then it keeps going. That's why the contractions get closer and closer together, because this positive feedback loop is getting closer and closer, because even when this is contracting, the signal back to the brain, the stimulative oxytocin, is continually being released closer and closer together, and those are causing the contractions to become closer and closer together. Talked about the fever. Any fever above 104 degrees Fahrenheit will increase the metabolic rate, digest more food. This produces more heat faster. Body temperature rises, further increasing the metabolic rate, and you see how it keeps going in one direction, and the cycle continues to reinforce itself, and then usually around 113 or 115 or 106 for some people, it can become fatal because proteins start to denature and the body starts breaking down. That's why people with severe fevers, you'll usually see them even in a hospital. They'll get cold packs and pack those cold packs on the patient to get that fever down immediately, and then start antibiotics and other stuff to try to help the issue that's causing the fever. But usually stop the fever first and then worry about what's causing the fever after that. So how do we start studying for this class? So medical terms. You can start looking at about 400 word elements. You can start combining the root, the prefix, the suffix. You can start seeing acronyms on how they're used. You can look at charts if you'd like that have the singular ending versus plural, so you can kind of go through all this. You can start seeing, okay, plural forms, they're not always easy. Ovary, ovary, ovaries, cortex, cortices, corpus, corpora, epidermis, epididymis. And then you start looking at these, you just get confused, get overwhelmed. So for most of you, unless you're a linguist that's fluent in Latin, Spanish, French, and a couple other languages, or maybe German, some Russian, the best way to do this is just to take a term one at a time. At the end of this lecture, I'll kind of go back and say, well, what did we just look at? What were some of the terms we talked about? And then I'll tell you why they're important. So for the majority, you're just going to learn what's an ovary. And then over time, you'll learn that ovaries. Most of you do know plural versus singular for that. But for some of the other ones, you're not going to know them until you start working in that individual field. Epididymis. Epididymis is skin. So dermatologists would care about this. If you're a nurse, you probably would never hear the plural of epididymis. These you probably will. Brachium is arm. Brachii, of the arm. So the biceps brachii is a two-headed muscle of the arm. You can have the antebrachium, which is the forearm. The brachium is the upper part. We'll look at that when we talk about the bones and the and muscles, because that's how scientific language is going to look at these. But for the most part, brachium, brachii, digits, and digitorum. Knowing some of these just while you're studying them, like you notice how I said brachium, we're not going to know until bones and muscles, so there's no reason to kind of worry about it right now. What we have left in this PowerPoint, we're looking at medical imaging. Medical imaging for this test coming up, exam 1, is what do we use it for? And what I want you to do while you're studying these, don't just study what is an X-ray, but what does an X-ray show you? So an X-ray uses some type of radiation. They change depending on what type of X-ray it is, but it uses radiation that will penetrate all different types of tissue, but bone will absorb it. So bone shows up as white. So most X-rays, when you look at it, you're pretty much just going to be looking at bones. Is something fractured? Is something damaged? And how quick is an X-ray? You pretty much put it on a table and it's relatively quick. If most of you have gone to the dentist where they put those little things in your mouth that's hooked up to a computer. They take a quick picture. They come back in and they have an X-ray of your entire teeth and uppers and lower. And it's very quick. So if you have a broken arm or a broken foot or any type of broken bone, they can get a quick X-ray, determine if it's broken, and then work on that. They don't need to set it up into any of these other types of systems or any types of imaging. That takes a lot more time. The reason why X-rays are still used, because they're quick. Now, radio-opaque substances. So this one's just the top of the brain, or top of the skull, looking at vessels in the brain. And with this one, you inject something that's opaque to X-rays. So you can do barium shake, you can do an isotope that will actually, show contrast, but pretty much anything that fills a hollow tube. So blood vessels or intestinal tract. If the blood vessel is leaking or blocked, radio-opaque substance will show that. If your intestinal has blockage or something like that, a radio-opaque substance will show that. If your intestinal tract has a leak or some issue where it's just not flowing properly, this is the type of imaging you would use because it shows how the tube kind of forms and how they go. If you saw a big blockage over here that was red when there was not supposed to be anything there, you might say that that was a blood clot or a stroke or something like that. So computer tomography or CT scan. It used to be called computer aided tomography. So computer aided was a CAT scan, now it's called CT, same thing. All this does is it uses x-rays, but it uses a computer to analyze the difference in that x-ray. So it increases the sharpness of the image, and one of the benefits of it is that it's not as much radiation, so it's not as dangerous to you. The drawback is that this is kind of limited because if you have a lot of water, it shows up as blue. So ventricles and stuff shows up as blue. Your eyes shows up as blue, and that because it penetrates it at a different speed, the computer can kind of add to that and say, oh, this part over here is all bone. This is a different type of tissue. That's a different type of tissue. But the pink in this picture would be bone. So you can see other things in more detail than you could with just an x-ray. These next two are pretty much the ones we're going to use in medical imaging. Positron emission tomography and a PET scan is different than a CT scan because this time we're not really using any type of x-ray to pick it up. This time you're injecting the radioactive substance into the body. So in a positron emission, you're actually injecting radioactively labeled glucose. And any part that is taking up this glucose means that it's active. Okay? So if it's using the glucose, it's active. So whatever's going on, like if you've had a stroke and you don't have a lot of, you don't have a lot of use in one part of the brain, a PET scan can pick that up. If you have damage in the brain, a PET scan can pick it up because it'll look more blue, it'll look more green, it won't be as active as other parts of the brain. So damaged tissues appear in a darker color. Using this kind of positron and emission, tomography, what the positrons are emitting, showing the use. MRIs is kind of the same thing, but this one's using a magnet. And the way this one does is this one uses water as its way of signaling what the tissue is. And kind of let me go over this. So what happens is if you change the magnetic poles, water will also change its magnetic poles. So water, north-south, we all live north-south, the magnetic poles. But if you change the magnetic poles via a huge magnet in this machine and then shut off that magnet, what happens is the water molecules will start to wobble back and forth. It'll always start to go back to north and south. And when it does that wobble, the denser the denser the tissue or the least amount of water will wobble less than if it has more water, it'll wobble more. And the computer, the scanner, actually will pick out different types of tissues and start to see how those tissues are composed. And if you're like, that's too much science in there, all you're looking at is a very good scan of the tissues based on how much water is in them. And if it's white, there's almost no water in them, that's going to be bone. The brain here, you can see it has more water, so it's a different color. And then you can use the computer in the system to pick up an optic nerve versus the muscles on the outside. So that MRIs uses water in a huge magnet. Sonography is something that's very popular at Palm Beach State because there's a program for it now, and this is usually used in childbirth because it uses sound wave. So sound waves and echoes, what happens is it sounds a frequency. The high frequency sound waves echo back from the internal organs, and then with the computer, this is a 3D scan. Most of them kind of look like that up there. But this right here, you can see growth of bones, you can see tissue growth, and you can see if things are progressing. And because it uses sound waves, the image is not very sharp, but for a lot of childbirth and obstetrics, you don't need to have something very sharp. All you're looking at is, are the bones growing? Is there a heart? Is there a nervous system? Stuff like that. So you can pick all this up through sonography. And then you have x-rays, bones, computer tomography. Takes more time. This is stuff you would look at if you wanted to see a more detailed x-ray. MRI, you can do functional MRI and see which tissues are being used. And if you're like, whoa, that sounds a lot like positron emission tomography. It is. So a lot of PET scans are being replaced by functional MRI, which just means there's a little F in front of MRI. And these are just looking at if tissues are being used. And that's a different type of view. And, bones, teeth, x-rays. Diagnostics of disease is CT scan. So if you want to see cancers, blood clots, heart disease, fractures, you look at CTs. MRI is non-invasive, no radiation because it's just using a magnet in the water that's already in your body. So aneurysms, muscle joints, looking at how your joints look and if the ligaments or muscles are going to be damaged. Stroke, brain tumors. Positron is going to be cancer, heart, cardiovascular condition, brain disorders, because it's looking at use of glucose. So it's really kind of good to kind of look at. And the problem with PET scans is that sometimes non-cancerous conditions can look very similar to cancerous conditions. So a lot of times they're just using more of MRIs to diagnose it in conjunction with the PET scan. And if you kind of review, you were doing the study guide while I was doing this lecture, and you remember the hierarchy of complexity from the textbook, from atoms all the way up to an organism, that's a learning outcome for this course. So you'll see, starting next chapter, when we start looking at the chemistry of life, everything up to that point until we get to the end of the book is an important learning outcome for you to memorize how an atom makes an organism work. So just because it's really small, those small components make a very large part of this of human anatomy. And then finally, the rest is going to be homeostasis, negative and positive feedbacks. So if you don't remember those different types of terms, go back. You're going to see them tested on the quiz. You're going to see them tested on the exam because they're learning objectives that are written on the first page of this of the home page for this class. And then other things, just kind of think about, when would I use these? Become the doctor, become the nurse. If somebody comes in with a break in the bone, what would you use? And just go through them and just try to think, what's the best way for me to diagnose a patient or for me to help a patient? And then good luck, and start with the reading and the assignments, and then you'll see me, or you'll hear from me again in chapter 2.